Okay, so for this set of audio lectures, it's going to be broken into two parts. The first part's going to be about B cell development, which is covered in Chapter 9, and then we have B cell activation, which is covered in Chapter 11. So same kind of setup as we had with the T cells, where we'll first talk about their development, and then we'll talk about their activation. So we want to keep in mind that for B cell development, that's going to happen in the bone marrow. It begins in the bone marrow, and then it actually gets completed in the peripheral. So it happens at the site of hematopoiesis, where we get the formation of all of the different cells that are going to be in the blood, one of those being the B cell. Okay. We also are going to get the selection where only negative selection is going to be required. There's no need to restrict to MHCs because the B cell receptor doesn't require the MHC in order to bind to antigen. Okay, so again, keeping in mind that T cells require those fragments to be processed and then presented on the MHC. B cells don't require that in order to get activation, so you don't need to go through that positive selection that we see in our T cells. Okay, so if we look at this figure on the left, we can see how we're going to have these early pro-B cells, and during that time we have our heavy chain undergoing recombination. Okay, and so that we are going to end up having that recombination of the VDJ heavy chain. This continues into the late pro-B cell. And then in our pre-B cell, that is when we're going to be having our pre-B cell receptor being expressed. And then it's going to undergo several rounds of cell division so that we then get the light chain recombination happening. So that then when we have our immature B cell, that's when we're going to have our B cell receptor selected for. Okay, so that we would have the heavy chain already confirmed because that was taken care of, if you will, during the pro-B form, and then during our pre-B form, that's when the light chain was selected for. Okay, so then again at the immature B cell, we would have our B cell receptor selected, picked out, and now we have to go through that negative selection so that we make sure that we're not binding any self, and that if we are binding self, that we go back and do some receptor editing. So again, during the immature B cell phase, this is where negative selection is going to occur and any kind of receptor editing that needs to occur so that when the cell then moves out into the peripheral, into the secondary lymphatic tissues like the spleen, that when it interacts with antigen, it's not interacting with self, it's interacting with foreign material or altered self so that we don't accidentally respond to that. So again, one of the things to keep in mind is the changes to the receptor during development. And so definitely this is where I would take some notes, some of the kind of general kind of pieces that you can kind of keep in mind is that pro-B, that the focus is the heavy chain. Okay. Pre-B is when you have the pre-B cell receptor, and remember that's where you have that surrogate light chain, so that to move to the next stage you have to select your light chain. So we have light chain recombination occurring during the pre-B phase, and then for immature B, that's where we have the negative selection and any kind of editing that needs to happen. The other thing you can see in this figure, again, focusing on the receptor, how the receptor changes so that in the pre-B, in the figure, you can see how you have the pre-B cell receptor, then it switches to the IgM expression. Again, this would be indicative of a B cell receptor that's functional. And then it's going to actually undergo class switching so that it's going to switch to IgD. Keep in mind that this is done by changing out the constant region that is going to be expressed. Okay, so this slide is just giving you, this figure is giving you some idea of where this progression is happening. And this is definitely something that's easier when I can kind of annotate or with the mouse kind of point out like, oh, do you see the hematopoietic stem cell? So hopefully you see that it's a little bit more, there's one off towards to the left, and then there's one also up near the osteoblast, near the bone. And so HSC is our hematopoietic stem cell, so you might remember that back from like our first unit. That's going to differentiate into a common lymphoid progenitor. So you can see how we have CLP, our common lymphoid progenitor. That's then going to differentiate into our pre-pro B cell. So it's not quite yet a pro B cell, so it needs to receive signals, and that's through the CXCL12, in order to then become that pro B cell. It's going to interact with IL-7 expressing cells, and that IL-7 expressing cells signals for it to undergo that selection of the heavy chain, so the heavy chain recombination. So it's going to express the pre-B cell receptor, again with that surrogate light chain. So we now have a pre-B cell. So if we traffic towards the right, we can see that. And then after it's gone through the light chain recombination, we have our immature B cell. And then that's going to get the signals in order for it to leave and become activated in the secondary lymphoid tissue. And then it can come back. So we can see this plasma cell coming back and interacting with the cells there in the bone. Okay. One of the things I didn't mention is that, you know, again, we have this hematopoietic stem cell. It's going to get interactions that, again, that will signal it to differentiate into that common lymphoid progenitor. Again, we get additional signals that are going to differentiate into that pro B cell, and then additional signals. So each step of the way, we're getting signals to kind of move to the next step, Okay. And obviously we can stain for those different signals. So in the bottom panel, what you're seeing is some of the staining for some of these markers. So like CXCL12, so those would be our green cells interacting with our B220 positive cells, and that would be our our pro B cell. Okay. With the IL on the panel to the right, we have our IL-7 positive cells, so that would be interacting with our pro B cell. So we want to keep in mind that the bone marrow structure is really dynamic and complex. And so in this figure, we're showing you here, we're in the bone marrow, and this cell is going to become this cell, and that cell is going to become that cell. But it's not a, you know, one-for-one-to-one linear path, and it's not they're all doing this at the same time. So obviously it's much more complicated, and there's many layers, if you will, to this, so that we do have our stem cells, right, that can differentiate and become different types of cells. And so we have the hematopoietic stem cell, and that's going to differentiate into, it's going to divide and then differentiate into the common lymphoid progenitor or the common myeloid progenitor. When we look at the lymphoid progenitor, some of those are going to become thymocytes, right? So we had talked about in the previous audio lectures about the T cell development. And so some of them are going to become thymocytes and go off to the thymus, but some of them are going to become that pre-pro B cell. And again, they're going to have the ability to divide. And so again, I just want to keep in mind that it's not like a one-for-one. Besides the stem cells and the cells that we're working on developing, we also have the stromal cells. And so those are going to be really important because they're going to provide the growth factors and the support that these cells need. So when we're seeing that, oh, this cell is expressing IL-7, that's going to be a stromal cell. That's going to be a support cell that it's going to allow that to happen. When we have CXCL12 being produced, when we have growth factors like that are going to be received, this is stem cell growth factor in which c-Kit is going to sense. Those are all being produced by these stromal cells, these support cells that allow for this development, and it's those signals from the stromal cells that are really important in this progression. So obviously if we had a stromal cell defect, that would then affect the ability to produce, move through this pathway to get mature, to first get to immature B cells, but then to get mature B cells so that we can actually have a response. So again, with hematopoiesis, we can define where we are in the stages based off of surface markers, also looking at transcription factors, and also looking at the Ig, the B cell receptor rearrangement that's occurred as an indication. So lots of different ways to experimentally look at where we are in the B cell development. So we can use antibodies that are going to mark the surface, and that's where we can do flow cytometry. So we can do cell sorting, where we actually will isolate the populations that are marked. We can also do things like immunofluorescence. So we can also do DNA analysis to look at what changes to the immunoglobulin, the Ig genes have happened, to determine whether a rearrangement has happened on the heavy chain and then on the light chain. And we can also use knockout mice or knock-in mice to determine the effects of different molecules on the pathway. So we could like knock out c-Kit, and obviously that would affect both our B cells and T cell development, or we could knock out IL-7 and look at its effect, or CXCL12. So again, we don't want to forget about that common lymphoid progenitor. So that's going to be that earliest step in lymphocyte development from the hematopoietic stem cell before we end up getting our B cell. So there's a number of different expression changes on the surface of these cells between the hematopoietic stem cell and the common lymphoid progenitor cell. And so you obviously could look at, these are some of the markers that you could look at to determine where you are in the stage. Just keep in mind with common lymphoid progenitors that they've lost that myeloid potential, but they do have the potential to become other cells. So they can become the thymocytes, they can become the B cell, but also remember in the thymus it is possible for the common for those thymocytes to differentiate into myeloid cells and other cells. It's not until they have they have notch expression that they have committed to the T cell lineage. Okay, so there is still some potential. When we have IL-7 receptor signaling, that's what really promotes cell survival and production of the early B cell factor, EBF1. And EBF1 would be, analogous to Notch. So Notch is what we look at in T cells to say, okay, we have like locked into this lineage. Early B cell factor 1 would be the similar. So when that is expressed, that is kind of the commitment step to becoming a B cell. This factor allows for the loci, the genes, to become more accessible so that they can be expressed. So again, there's a number of different transcription factors that are involved, signaling factors that are involved that we can look at to determine whether we've activated certain pathways that are necessary in order to form our B cell. So we'll be talking about some of these different factors, but you can see how here with the the IL-7 pathway, we're going to have the receptor present. We're going to get JAK1, JAK3 activation. That's going to lead to STAT5 phosphorylation and translocation to the nucleus, so that we're going to get the EBF1 activation. And that's going to lead to the activation of a number of other factors, all of which are kind of associated with B cell survival, proliferation, and development. So when we look at the different markers, we can use those in order to classify which stage of development we are in. So we had mentioned about in that figure how we had that pre-pro B cell, we have that, and then we have like an early and late pro B cell. And so for those, we can look at markers like RAG1 and 2 expression. Again, that IL-7 receptor expression is really important, and that actually decreases once we have our, once we're further along in our B cell receptor process. So you can see how that changes, so that when we have our pre-B cell receptor being expressed, and as we move through into our second checkpoint, being the immature B cell, that expression is lost. One of the markers that is held throughout is B220, which is CD45 receptor, so that it can sense CD45. Okay. Another marker that isn't expressed early but then gets expressed is CD19. Okay. And so CD19 in that pre-pro and early pro B cell is not expressed. Okay. Again, if we keep in mind what's happening with the B cell receptor genes, the Ig genes, that really helps kind of differentiate the different stages, and so that's what I would kind of focus more so on. Keep in mind that in the earlier stages we have our germline heavy and light chain, so we haven't made any kind of changes to that, right? So that would be our pre-pro B cell. As we move into being a pro B cell, that's when we're going to have our heavy chain starting to be altered. And first we're going to do DJ, then we're going to have our variable being selected for. Once we have that occurring, right, so now we've selected for the heavy chain, that's when we're going to move into the pre B cell. So we have a pre-B cell receptor that's being expressed. Remember that our surrogate light chain would be expressed with that. So then when we move to our next step, and this would be the small pre-B cell, that's when we have the light chain rearrangement occurring. So then we're not expressing the pre-B cell receptor, we're starting to express the actual B cell receptor. And so at that point, when we're expressing the actual B cell receptor, which is IgM, at that point we have undergone both heavy and light chain arrangement, and we would be an immature B cell. Okay, so we tend to use the basal nomenclature for naming of these different stages, and again, that would be what I was just talking about, the pre-pro, pro, pre-B, immature. Okay. There is another nomenclature which is known as Hardy, and that goes through an A, B, C, C prime, D, E. Okay. So again, that's not commonly used, but it you might see that, and in some ways it might seem like that's a simpler nomenclature to use. But again, I think the basal one with the pre-pro gives you the idea of where you're moving through in the progression. So you're not quite yet a pro B cell because you haven't got your ducks in a row. So you need to get those taken care of. So you need to start selecting your heavy chain, right? So there's something that's unique to that before you move through. So this page just summarizes a lot of what I was just talking about. So I'm not going to go through this again, but definitely you should know some of the key markers of the different stages. So key characteristics. So, that expression of B22 is not found in the common lymphoid progenitor. So it's something unique that would indicate that we do have that B cell lineage occurring, right? So that we're going to start to, this is a pre-pro B cell. It is going to become a B cell if it successfully makes it through all of the steps, right? So again, I would definitely go through this and kind of make sure that you understand again the sequence, the staging of the different B cells in their development. So for the immature B cell in the bone marrow, it has to go through tolerance. So it has to be checked to make sure that it's not actually going to respond to self. So we want to make sure that we delete any kind of self before it's allowed to go into the peripheral tissue. So for that immature B cell, it can undergo clonal deletion when it gets selected for this selection. It could have the receptor editing occurring where we could rearrange the light chain. So we go back into the recombination machinery, rearrange and edit the light chain so that we could end up with a different B cell receptor, a different IgM, so that maybe that would not actually respond to self. Okay. And then lastly, we could also get an anergy, so it could become anergic where it wouldn't respond to it. So for the immature B cell, the surface markers it would have would be the IgM, right? So it would have the B cell receptor. It would have B220, CD25, IL-7 receptor, and CD19. So we could use any combination of these markers to identify, well, not any combination. You would need this combination in order to identify an immature B cell versus like another stage, okay? Because obviously some of these markers a mature B cell would have, right? So again, during this process, we're going to get that selection. Again, so it's going to be tested. So again, negative selection, so because we do not want it to react to self. So with the negative selection, it is a way to try to eliminate as many self-reactive B cells as possible. It does not eliminate all of them. So there are some self-reactive B cells that are allowed to go out into circulation. But again, the idea is that these stromal cells of the bone marrow would end up producing, just like we saw in the thymus, they would be producing all these different proteins, and then they would present them on MHCs, and then our immature B cells would be able to interact with them. And if they did recognize them, then again, this is where we can have those different outcomes where the B cell would not be allowed out into the peripheral. It would actually get deleted, so clonal deletion, or this is where we would undergo light chain editing occurring, or it wouldn't receive any signal, and so it would become anergic. The ideal outcome is that everything's right, and that the B cells then becomes mature and goes out into the peripheral. So in the figure, you can see how we have from the bone marrow, we have that immature B cell moving into the spleen. And when it moves into the spleen, we would then differentiate it into two different subsets, T1 and T2. So just keep in mind, these are B cells, they are not T cells, even though T is in there. And so there's going to be gene expression changes as it further matures in the spleen. So, we're going to go from the T1 stage to the T2 stage. Then we're going to have that mature B cell moving into the follicle. and then it can end up leaving so that it can go off into the peripheral. So we'll look a little bit more at this T1 and versus T2 stage. So when we're looking at these subsets, T1 subset is still going to be undergoing screening. So it underwent negative selection in the bone marrow, but we want to, just in case something went out, because as we mentioned, not all self-reactive B cells are eliminated in the bone marrow. So we want to go through another round of the selection. So T1 subset is going to undergo another negative selection event, so that if it does respond to self, again, it would be deleted out. Once it passed through this negative selection, it moves to the follicle, and so that's where we're going to have the T2 stage. Okay, at that point, what we're looking at is the B cell receptor strength, and so we want to select for ones that have a strong B cell receptor signaling. So what you do is you end up with BAFF binding, and if we don't get that signal, we're going to end up getting cell death. If we do, then that's when it would be selected for and would become a mature B cell. So when we look at the response of the B cell receptor in these different subsets, so our T1, our T2, and also our mature B cell, they can have different responses to the B cell signaling. And again, this is what's going to help them move through to the different stages of development. So for our mature B cells, the first stage that they are in is a primary B2 B cell. So they're going to move to lymphoid follicles. And so at this point, they're going to express high levels of both IgM and IgD on their surface. So you can kind of think this is their old B cell receptor and then their new B cell receptor. Because again, keep in mind that the difference between these are the constant region, which constant region segment is being expressed. And so they can actually express both of them, okay? They are able to circulate into the blood and then back into lymphoid organs. And so this actually helps to have them respond to antigens because they'll receive T cell help to produce their antibody. So once they move into the peripheral, they have a half-life about 4.5 months. So they're, if we think about this like the lifespan of an average cell, it's a pretty long lifespan. There's a number of surface markers that can be used to distinguish between them. So again, the expression of IgM versus IgD is the primary way in which you would be able to distinguish between them, but there's also some of the CDs, clusters of differentiation, that are also unique to the different stages. So when we look at B cell and T cell development, there are many characteristics that they actually share. So they both are going to undergo rearrangement of gene segments. They both undergo screening process to avoid those cells that are reactive to further develop. And then also when we look at the production of the B cells and T cells through development, there are small subsets that have specialized functions, but there's also the larger general purpose subset also that is being produced. There are some differences between T cell and B cell development. So obviously location is going to be different with B cells. That development is happening in the bone marrow and then further in the spleen. With T cells, your development is happening in the thymus and screening is occurring there too. Again, the screening processes are different. we have the positive and negative selection in T cells, but only negative selection in B cells. Keep in mind that positive selection in the T cells is where we're selecting for MHC recognition. And again, in B cells, we don't have to do that because B cells don't require the MHC in order to see its antigen. Okay. The specific arrangements in gene segments that are being utilized obviously are different. And then the overall outcome of the antigen receptor simulation is going to be different, right? So again, T cells require presentation and differentiation into helper and killer subsets, where B cells are going to require T cell help to secrete antibodies. So again, there's a little how they get to their function and what their function is unique to them. So this figure that's on the next two slides, or this table on the next two slides, just goes over some of the comparisons between the T cells and B cells in a little bit more detail. That previous slide does a nice job of, the previous two slides do a nice job of summarizing up so that you don't have to look at every single row. And again, like obviously things like location are going to be different, how they start out as the hematopoietic stem cell. Obviously in the bone marrow is going to be the same, so they both would have those, but when we talk about development continuing to the thymus, that is unique to the T cells. Okay, a lot of similarity between the receptors and they're being selected for. Obviously, you know, the genes that are the heavy chain versus and beta chain for T cells are going to be slightly different, but the overall process in development is going to be similar, where you have your pre B cell receptor and you have a pre T cell receptor, and then when we look at then their light chains, there's going to be light chains. You know, there's going to be genes that code for chains that are similar to light chains in both our B cell and our T cell. Okay. Then keep in mind that the selection process is a little different because T cells have to go through that selection process for MHCs, where we don't have to do that for the B cells. So just in summary, again, I think those two slides prior to that table do a nice job of summarizing similarities and differences. But just in summary, B cell development pathways are similar to T cells pathways, and just like that, they're complex. There's a lot of little nuances to it because what's being upregulated, what's being downregulated. But definitely understanding B cell pathways helps also understand T cell pathways. So again, I would certainly recommend kind of thinking about comparing and contrasting and understanding those really key similarities and differences to help to understand them. Okay, and as always, if you have any questions, you can always contact me.